Hey, this is Russ Altman from The Future of Everything. The holidays are here, and with that, many of us are preparing to enjoy meals with family and friends. One of my previous guests on the show, Professor Michael Fischbach, told us that the gut biome, that is the complex community of bacteria that lives in our gastrointestinal tract, is what makes digesting and extracting nutrients from those meals possible. It's a fascinating conversation about the important role that bacteria play in the human digestive system. I hope you'll take another listen and enjoy. Before we jump into this episode, I'd like to ask you to rate and review the podcast. It'll help others figure out if they're interested in the future of everything. Now, in the last few decades, it has become clear that the bacteria that live in our body, particularly in our digestive system, stomach, intestines, what have you, play a critical role in our health and disease. We hear about the microbiome, the set of bacteria that are living within our bodies that form this little ecosystem. They interact with one another, and presumably they interact with us. The word biome was originally used to describe all the living things in an environment. The mountains, fields, the polar ice caps. The surface of the Earth is the largest known biome, at least the largest known to me. Microbiome then refers to the tiny little environments inside our body that host a variety of living organisms, mostly bacteria. Now, when we hear the word bacteria, we often think of infections and disease, and indeed some bacteria can cause disease and do. But when we are healthy and feeling good, there are still millions of bacteria from hundreds of bacterial strains or species that are living and thriving within our body. They colonize our colon very quickly after birth, and they seem to be critically important for helping our immune system develop, helping us digest some sorts of food, responding to environmental chemicals, and other things. If we disrupt our microbiome, for example, by taking antibiotics for a strep throat or a urinary tract infection, the antibiotics kill the bacteria that are causing the infection, but they can also mess up this little ecosystem of friendly bacteria that are happily living in our intestines. There can be noticeable changes. Definitely our digestive system can change, and maybe our immune system and other elements of our health. Many of us have been told to keep our microbiome healthy. Well, what does that even mean? What am I supposed to do? We should have a diet rich in fiber, fruits, vegetables. Okay, that sounds okay. Maybe have yogurts and other sources like lactobacillus that seem to be part of a normally functioning, healthy microbiome. Several years ago, a prominent medical journal reported that the eating habits that were associated with healthier outcomes, less heart disease, less arthritis, less diabetes. And one of the foods that was at the top of the list was yogurt. A colleague of mine who works at MIT had two dogs. They were siblings, and he decided to do a trial where he fed one dog a diet supplemented with lots of yogurt, and the other didn't get the yogurt. And he said that within a few weeks, the fur, the coat of the dog that was yogurt eating was noticeably thicker, shinier, looked beautiful. And then the sibling dog had kind of a normal coat of fur. So after visiting him, I told this story to my family, and my children immediately started eating large amounts of yogurt, seeking the thick, shiny, healthy hair that blessed the yogurt-eating dog. So was that a microbiome-mediated cosmetic improvement? I don't know. But our understanding of the relationship between our microbiome and health is in its infancy. We know very little about the mechanisms by which bacteria living within our bodies affect our health. My guest today is Dr. Michael Fischbach, an associate professor of bioengineering and an expert at studying the microbiome, the bacteria that live together, the chemicals they make and consume, and the ways in which this may affect human health for better or worse. Michael, bacteria are like little factories that make and release all types of chemicals. Your lab studies these chemicals. Are they helping us, hurting us, or we have no idea?
The easy answer is we have no idea.
Great. Let's have a conversation.
Since this is a show about the future, very likely in the future what we'll understand is that the answer is all the above. And the way we can understand the chemical aspect of this is that multicellular organisms have all made a compromise with bacteria. In fact, there's no way to live on the planet without being coated. You can think of yourself as a cylinder, on the outside and the inside surface of the cylinder slathered with bacteria.
A lovely image.
A lovely image. And one of the interesting things about looking at the digestive tracts of all sorts of different animals that we all have learned about as kids. So if you look in their insides, their digestive tracts have completely different morphologies. They look entirely different one from the next. And you could imagine, although this hasn't been proven, that the tube length and shape and composition has been optimized for extracting nutrients from food, except it's not the tube itself that's extracting anything. It's the bacteria living inside it that we're using in the sense of a chemical reactor to break down the food and make its nutrients accessible for us after the bacteria have had a chance to chew.
So these bacteria are absolutely critical for our digestion. And,
they are.
And in fact, if we didn't have them, are there certain kinds of foods that would just pass right through that they are absolutely mandatory for the digestion of?
By all means, and the best example of that is fiber. So if you eat fiber, you cannot extract any of the sugars in fiber because they're tied up in a way that makes them difficult for your stomach to unpack. Bacteria in your gut, on the other hand, can chew right through them and turn the carbon in the fiber into acetic acid and propionic acid, things that rush right into the cells in your intestine and which use them as an energy source. So
that's amazing. And that suggests, therefore, that when we do take an antibiotic for some sort of infection, we really might, for example, be abolishing our ability for that week to digest fiber. Are these bacteria just as sensitive to antibiotics as the bacteria that we're trying to kill when we have an infection?
They are. And the interesting thing about antibiotics is that our colleague David Relman at Stanford did the pioneering work to show is that if you give a healthy person a course of antibiotics, they may end up after the course of antibiotics, during the time when they have an antibiotic on board, the composition, the roster of bacteria in their gut changes dramatically. They may, what he showed, in the end come back to a composition that looks much like where they were before, or they can end up in a completely different place. And so this stochasticity of what happens following a course of antibiotics is one of the most interesting things that's come to light to date around the microbiome. That means you really don't know what's going to happen.
So I know that one of the things that your lab looks at is not just the breakdown products of the food that we eat, but these bacteria, I believe, are also making new molecules and secreting them into your gut, and they might be interacting with your health. What's known about that?
The answer is, again, not very much, but it's good to set the stage. What really happens when you eat a piece of meat or a salad is that much of that carbon ends up in the small and the large intestine. It's not absorbed higher up in the stomach. And so the bacteria are really getting the first chance to chew on the food. They turn it into, and this is the interesting part, dozens, maybe hundreds of chemicals that end up in your bloodstream at levels that are similar to what a drug achieves in your bloodstream. So your blood is a cocktail of chemicals that result from the breakdown of food by bacteria. We know very little about what any one or another of them is doing.
This is The Future of Everything. I'm Russ Altman. I'm speaking with Dr. Michael Fischbach about the little bacteria that live in your gut. And now what we just heard is they not only make small molecule chemicals, but these chemicals get into your bloodstream and achieve levels that could be similar to the levels in your blood. Do we know if they have the kinds of effects that drugs have on our body? Does our heart rate change? Does our immune system respond? Or if we don't know, how would we ever find out?
Yeah, good question. So one good example to a story to relate is that a guy named Stan Hazen, who's in charge of preventive cardiology at the Cleveland Clinic, a place that is a basin that catches all sorts of patients interested in heart disease or maybe at risk, he became frustrated that the marker people use to measure heart disease risk, called C-reactive protein, doesn't work very well. It works pretty well, but not very well. So he decided he was going to look systematically through blood for chemicals whose levels were a better predictor of cardiovascular disease risk. And lo and behold, he found that a chemical with a very unwieldy name, trimethylamine N-oxide, which people abbreviate TMAO, was a much better predictor of your risk of having a cardiovascular event, especially a thrombotic event where your blood clots more than it should.
Yes, this is bad.
This is bad. And that chemical happens to derive exclusively from gut bacteria. So this has led him on a very interesting journey to understand why are bacteria making the precursor to TMAO and what exactly is TMAO doing that's harmful. And
Yes, please. So we don't know if TMAO is the troublemaker or is just like a sign that there's trouble, or do we?
So Stan thinks that TMAO itself, that there's good evidence that it may be the troublemaker. It may make platelets, blood cells, sticky and cause them to clot more than they should. Stan did an interesting study that may suggest a solution to the problem, which is that he managed to convince a group of vegans who
are, by the way, hard to convince of many things.
Vegans are strong-willed, as we know. If an omnivore like you or me eats a steak, then we'll see shortly thereafter a spike of TMAO in our bloodstream because the bacteria were chewing on the carnitine and the choline in the steak, and that ends up becoming TMAO.
Okay.
If a vegan eats a steak, which he convinced them to do, it turns out they make no TMAO from it because they lack the bacteria in their gut.
The bacteria kind of died out or left town because there was no meat to eat.
No niche for them.
And the vegan bacteria don't make TMAO, evidently.
Not at all. And so that would suggest that there is a way to have a healthy gut population, gut community that doesn't make any TMAO. So you and me may in the future be able to eat steak without suffering the ill effects of TMAO by changing the composition of our gut community.
And this is great because I know that another thing that you do in your work is kind of engineer the bacteria. It's not just let's study them and watch and see what they do, but you're a little bit of an interventionist. So in what ways can we intervene in a microbiome?
So that's a good question. We, continuing the thread, began working with Stan, and we took a bacterium that is a robust producer of the precursor to TMAO and deleted the gene that was responsible for producing it. Stan then put these bacteria into mice in his lab that were germ-free, had no bacteria in them to begin with. And
so you can control what they're getting.
So we can control what they're getting and put them under conditions where they would be prone to having an atherosclerotic event, and showed that just because that these two mice that were identical save for one gene in the genome of one bacterium in their gut behaved completely differently in terms of their risk of having atherosclerosis. The mice that don't have any bacteria making TMAO in their guts were completely fine, whereas the ones that had a TMAO producing bacterium in their gut got sick, got cardiovascular disease.
Unbelievable. So here's a question is, why would these bacteria do this to us? So here we are hosting them in our intestines. Welcome to my intestine. Why are they making? What's the advantage to the bacteria, if any, to make the TMAO and cause us, its host, all kinds of problems?
So here I'm going to try to make something highly esoteric sound interesting on the radio.
Okay, let's do it.
The answer is that bacteria in the gut have an extraordinary problem, which is that there's no oxygen in the gut. You and I take for granted that there's oxygen in the air, and so do normal bacteria that live outside the gut. They use oxygen to run their battery. Oxygen turns out to be great at having electrons dumped into it, and as you know if you know anything about batteries, that electrons go from one place to the other, and so the sink for the electrons is absolutely critical. Bacteria in the gut have this problem. There's no oxygen. They have to find another sink, and it turns out that things in food are not just great sinks, they're great sources. So they use, instead of oxygen, chemicals as sources and sinks for electrons to run their battery, and we happen to live in their bus exhaust. The chemicals they make wash into our bloodstream because there's nowhere else for them to go.
So this is a byproduct of them just trying to get by.
Exactly.
Now let's go back to your experiment with the two mice, where one of the mice you deleted the ability to make TMAO. Did that in some way mess up their battery?
It didn't, and the reason we think it didn't mess up their battery in this instance is that bacteria are wily and crafty. And this bacterium, Clostridium sporogenes, that makes trimethylamine, the precursor to TMAO, has other options. It's good to have other options in this world, and Clostridium sporogenes has other places it can dump those electrons. So in this instance, we don't think we think that the reason why Clostridium sporogenes didn't suffer was that it had another place it could dump its electrons.
So this, going back to my question about can we engineer these guys, the answer is maybe we can, and maybe we can have our steak and eat it too, or something like that with these engineered bacteria.
Exactly.
Now, I know that another thing that you think about engineering is the actual community. So it's not that our intestine is just filled with that one virtually unpronounceable bacteria that you just mentioned a couple of seconds ago, but that they live in these kind of very interconnected communities. What do we know about those communities, and are they fragile or robust? Like, do you have to have a certain neighbor, or is it optional? And how might this influence our health, and how might this be an engineering target for you and your colleagues?
Excellent question. So I'll motivate this by saying that the entire history of microbiology and human research is that bugs have been studied one at a time. This is a way that we've learned almost everything we know about how bacterial cells function.
And when you say bugs, we're talking bacteria.
We're talking bacteria,
okay.
and I'll be more disciplined for the rest of the talk. However, the way they live in the real world is in the context, as you point out, of complex multi-species communities. And so we know very little of how this works. I'll motivate all this by telling you another story, which is that there's a guy named Josbert Keller, who is a physician in the Netherlands, a very unassuming guy, who had for a long time been seeing, he was a gastroenterologist and had been seeing patients who had difficult infections in their gut that were refractory to treatment. And instead of the normal treatment that a heavy-duty antibiotic that you're usually given for this that has a low rate of success, he had been taking fecal samples from healthy people, suspending them in saline solution, and putting them through a tube that goes in the nose and down into the stomach, into the gut of the infected person, with a high rate of success. So
wow.
This is called a fecal transplant.
Yes.
So Josbert decided to do something interesting, which is that he and his colleagues knew that this worked quite well. But nobody else was going to be convinced of this unless he did the gold standard of clinical trials called a randomized controlled trial. So he set up such a trial, was very small, had only roughly 15 people per arm.
And you have to recruit people to receive the treatment, so to speak.
To receive
which could be controversial and a difficult decision.
That's right. So
although if you're a desperate patient with a terrible infection, you might be willing.
In his telling, patients were lining up for this because they had heard by word of mouth how successful it was. And so what happened was, in the middle of his trial, he had to stop it early because it was working so well. 14 out of the 15 people who received the treatment got better immediately, whereas only 4 out of the 15 people who received the antibiotic got better.
And then it becomes unethical to continue the treatment, to continue the trial, because it's pretty obvious that it's working.
That's right. And what was so interesting about this is that it ended up being published in the New England Journal of Medicine, the journal where only the most interesting, amazing results are published, and set off a storm of activity. And now, at many large academic medical centers, just a few years later, if you come in with an infection like this, you'll no longer receive antibiotics. You'll receive a fecal transplant.
And I guess it almost goes without saying that the fecal transplant is not giving a single agent. It's the entire mix of bacteria in the healthy donors, I guess you would call them. And that's why it takes a village to solve this infection. And it's not just about one bacteria that's missing or needs to be added back.
That's right. This is the community as the drug.
One of the things that you think about is manipulating these communities, both for discovery and maybe for interventions related to health. How do you manipulate a microbial community, and why would you want to do that?
That's a good question. There are crude tools you can use, and then there's a solution that we've begun working on that I'm very excited about. Going top down, taking an intact bacterial community from the human gut, which may have many hundreds to low thousands of species, and doing anything specific to it is a daunting task. There are crude things you can do, like using antibiotics or like using chloroform to kill off everything but the spores. And people have made interesting progress using approaches like that. But something that's got us quite interested is the prospect of, could we build one from scratch?
Now, before we go to that, the daunting part of it, is that because it's so robust that if you try to make little changes, it just goes back to how it was? Or I can imagine it being daunting in many ways. So give me one more sentence about why that is so hard.
It's daunting because there's no way to do anything precisely. There is this property of communities that we all understand but don't get why it works, which is their incredible stability and resistance to all but the most profound perturbations. And so figuring out a way to take one bacterium out of a community is something people haven't yet figured out.
Gotcha. So instead, what do we do?
So instead, there have been early efforts to build communities from scratch, but they've topped out at 15 to 20 or 30 bacterial species. And that's been a lot to mix together and try to see any emergent properties like the ones we just discussed, stability and resistance to perturbations. And so we've begun trying to build, even before we understand them, extraordinarily complex communities that have more than 100 bacterial species in them. This is work we haven't published yet, so all the listeners should take
we heard it here first.
We heard it here
literally.
First, and take it with an enormous grain of salt.
Has not been peer-reviewed yet.
Has not been peer-reviewed yet. One of the interesting things, though, is that you asked the question, are they working in harmony, or are they fighting with each other, or what would characterize the way that they're interacting with each other? And one of the early hints that we've had is that I had thought the way that ecosystems work, which we all learn as a child, is what I call the Lion King model, where everybody depends on everybody else, and if you remove one animal or bug or plant from an ecosystem, the whole thing is going to crash.
Yes.
The 100 bacterial species we've mixed together seem not to behave that way. If we take one of them out at a time, in fact, almost everybody else doesn't care, except for a few friends. And the few friends care a lot.
And it rocks their world.
So we think that the way that this ecosystem is structured is in small groups of organisms that care a lot about each other, and everybody else could care less.
Did you design it to be that way, or did you kind of get lucky?
Not at all. We designed it to look a lot like a normal human gut. We took bacteria that were the most prevalent, some of them very abundant, some of them at 1 in a million in the community, but every one of them is present in almost every person. So we mixed them all together, had no idea what was going to happen, and it turns out that they're quite stable together.
Now, as you get more and more successful at building these communities, what is that going to enable? What should we be excited about is going to let you discover or learn or intervene?
The fecal transplant we talked about before, which has been so amazingly effective at curing these difficult gut infections, hasn't worked so well for other diseases. And the presumption is that that's because you may be able to cure another microbiome-linked disease like Crohn's or colitis or like obesity using a fecal transplant, but not just anybody's feces is going to work. We need a very particular group of bacteria. And so we're hoping that this enables us to design in the same way that you can nip and tuck and make small changes to a chemical or to an antibody therapy. We think this is going to enable us to do that for bacterial communities.
So precision fecal transplants.
Exactly.
Don't just put back a kind of otherwise normal person's, because that's not enough, but understand this diabetic is going to have these problems in their microbiome. We have to surgically introduce these new, and it probably won't be single bacteria. It'll be these little groups that you were referring to.
In fact, that's right. So we think that we're going to mix together a group of 100 to 200 bacterial species, and that we're going to be able to precisely control what chemicals they make or how they tickle our immune systems, and that's going to be unusually useful.
So in order to do a therapy, you need to make a diagnosis. In the future, are we going to be providing samples of our microbiome on a routine basis to our physician so they can kind of assess our health, diagnose disease, learn about us in ways that they now do with blood tests that come out of our arm?
We think definitely. Where we are right now is that you can look at the roster of bacteria in somebody's gut and learn a little bit, but not a lot. The little bit you can learn is that, for instance, patients who have Crohn's disease, their gut communities look wildly different generally than a healthy person. And so if you took a 10-year-old and showed them data, just community rosters from one group and another group, one of which was healthy people and the other was Crohn's disease, they would very easily pick out that there's something amiss.
And the Crohn's disease folks look alike? Do they look like one another so that there's kind of a look for Crohn's disease?
In fact, they don't. There are sort of, I would roughly define it based on recently published data as 4 or 5 buckets, where within a bucket, they do look alike. And each of those buckets look a lot different from the very big, messy, healthy bucket of people.
So that sounds valuable to me, because that means maybe there's not just one treatment for Crohn's disease, but maybe there's 5 treatments, and that the marker for which of the 5 you should get is how your microbiome is shaping up.
That's right. And so here we're talking about diagnostics, and so I do think that a diagnostic for Crohn's disease is not far off, which would be much simpler than going in for a colonoscopy. This could be an early flag that sends somebody in for a colonoscopy. But I'd also point out that there is a debate in the field about whether the change in the microbiome precedes or is a consequence of the disease.
I was just going to ask you that, because the real value would be if these are early detectable things before we see frank disease with lots of symptoms. And I guess the jury's out on this right now?
The jury's out, although I would say that a point of view that's emerging is that the way Crohn's disease probably works and lots of other diseases, so this is important to discuss, is that it's probably the case that you have first a host genetic predisposition, but that itself is not enough to trigger disease. You need a second event, which is a change in the composition, the roster of the bacteria in the gut, and the two of those together are what give you disease. So it may not be that talking about this as cause or consequence is appropriate. It could be that there's a multi-step chain of events, and the change in the bacteria is somewhere in the middle, but is necessary for getting disease.
And that would certainly explain why we see people who have genetic risks, but some people get the disease and others don't, and we never really understand why. And people kind of throw their hands up and say, well, it must be some environmental factor. A huge environmental factor here is going to be the bacteria and the signals that they're sending to the host, which is the human.
Exactly. And moreover, wouldn't you like to look at people who are genetically predisposed but don't have disease? These people must be colonized by bacteria that are somehow protective. The trigger has not happened in them.
So I'm sure I am wondering, and I'm sure we all are wondering, what about this lactobacillus yogurt eating? I made my introductory comments about this. Is there scientific basis for diet modifications of this type as potentially healthy, or is it really not science-based yet?
Fermented foods, there's enough evidence to suggest that they're probably good for you, but I would be skeptical about anything more than a mild statement to that effect. My guess is that since the bacteria in yogurt, for example, and other fermented foods are only temporary residents of the gut, they only spend 2 or 3 days there and then they're gone, that any effect they have is going to be transient, and it's not clear to me how much of an effect it is. My guess is that in the future, we're going to be putting bacteria in our gut that sit and stay for a long time, and that's going to be interesting.
The one final thing I wanted to ask you about, because it's been in the news, is the idea that there is an old person microbiome and a young person microbiome. We've seen, I think there's been stories about old mice that get a transplant from young mice and they do better. So what's known about these aging effects of the microbiome? And it sounds like it's not a stable thing. Even if you're in the same house eating the same diet, it sounds like there might still be changes over the course of your life.
Very early days in this, but that might be the case. My guess is that all of the effects we're talking about now, let's say 1/3 of them turn out to be true and the other 2/3 turn out to have been spurious. But the more important point is that in the future, I doubt we're going to leave the composition of our microbiome to chance. We are going to program it. And so all of these things we're discovering now, we'll also discover that there's an optimal composition of the microbiome for somebody who's 60 years old, and they will get it.
You have been listening to the Future of Everything podcast with Russ Altman. I want to remind you that the Future of Everything started out as a radio show on SiriusXM, so you'll hear references to that. Now it is 100% podcast. We have more than 200 interviews in our archive, and they cover an extraordinary range of topics. If you're enjoying the podcast, please consider subscribing or following so that you can be alerted to every new episode and never be surprised by the future. Maybe tell your friends about it too. Definitely consider rating and reviewing it. That helps us grow, improve, and also spreads the word. You can connect with me on Twitter at @RBAltman and with Stanford Engineering at StanfordENG.